In the last video we were introduced to DNA. Now we're going to learn some more about it. So by the time this video is over, you'll be able to define the words that you see on the screen, explain why DNA has to be replicated or copied, and why cells divide. DNA is found in the nucleus, as we know from previous sections. How much DNA do you think that you have? You actually have 6 feet. Think of a person who's 6 foot tall. 6 feet of DNA per cell, and cells are tiny. So how do we make 6 feet of DNA fit? We have to wind it up. And you can see in the picture that we use special proteins and different other molecules to help wind up DNA so it will fit inside all of your cells. Now, sometimes we need to make more cells, and we'll go through those reasons shortly. In order to make more cells, we need to make more DNA. So, why would we want to make more cells? Well, the first reason you can see in the left picture. That would be to grow. In a previous unit, we talked about how cells can't just expand. You can't just have the plant there be a couple cells that expand in size because of that surface area to volume ratio. What they need to do is they need to make more cells. So mitosis is the process of making more cells for growth and also for repair. Those are the two things you should write down in this section of your notes for mitosis. Repair, think about if you had a cut and you didn't ever make more cells to fill in that cut, you'd be full of gaps by now, thinking about how much kids get hurt when they're little. If those things never were able to fix themselves, you would be a wreck compared to what your skin looks like today. So mitosis is for growth and repair. Meiosis is another cell process, and we'll talk about these later on in this unit. But meiosis, the point of that, making more cells, is to create a new organism. When we're making new cells, something that's really important to understand is that the DNA never leaves the nucleus. It always stays inside. So think about this. Why would you not want to take DNA out of the nucleus when you make a copy of it? Imagine that you had a safety deposit box with your social security card, your birth certificate, your credit cards, all of the most important information about you. And you would never want to take it outside of that box because if you did, there's a chance that somebody could actually steal it and mess it up. Similar idea within cells. Because DNA codes for everything, we don't want to take it out because we don't want to screw it up. When we need to make more DNA, there's a couple different steps. So let's go through these basic steps. First, DNA gets unzipped by a molecule called helicase. Helicase, anytime you see ACE, it's an enzyme which speeds up a reaction. So helicase is going to come in. Remember, DNA in this picture is 2 strands, double-stranded, and helicase comes in and breaks these hydrogen bonds between the A's, T's, G's, and C's and unzips the molecule. DNA polymerase then comes in, and what you're looking at is one strand right here, and DNA polymerase is forming a new strand. It adds new nucleotides, which are those PSNB things, to each side. And you can see in this picture the DNA gets unzipped. You have 2 sides. DNA polymerase is making a new strand on each side. DNA is then proofread, and each strand is glued together, so it's held in place. And if there's any mistakes, then we usually catch them. However, if there are mistakes that are missed when DNA gets proofread, those mistakes get read over and over and over. So you can see in this bottom piece of DNA here, there's a mutation. Instead of being purple like the correct copy, now that there's this red copy, that's going to be copied from generation to generation, cell to cell, and that can be passed on genetically and then cause disease. I'm going to show you an animation because sometimes these processes are hard to imagine. So here we're going to copy DNA replication. We have various molecules. We talked about helicase and polymerase. You don't really need to worry too much about the other ones. But here we go. Here comes helicase, and it's unzipping the DNA. DNA polymerase, we have 2 different ones that are kind of working. You see a red molecule shooting in and out. DNA polymerase is working on each strand to copy that strand and bring in your free nucleotides, your PSNBs, to make another identical copy. So this original strand is getting unzipped, and it's used as a template to make 2 brand new strands. The pieces get glued together and proofread to make sure that there's no mistakes, and voila, you end up with 2 new pieces of DNA that were an exact copy of what you started with. When a cell is not dividing, and it won't be anytime soon, the DNA, all 6 feet, it looks like this. It's called chromatin. I like to call it the spaghetti form because it really looks like a bowl of spaghetti. In chromatin, DNA is resting. It's uncoiled. It's 6 feet long. It's kind of a mess. You wouldn't want to try and copy that and make a new copy because it would just be awful. So what we do is we wind up that DNA. Let me go back a slide. We wind it up on those proteins. We make it this nice X shape, and that's called a chromosome. Chromosomes are a lot easier to move than chromatin. A chromosome gets wound up, the DNA gets wound up on what's called a histone. That's that protein that DNA winds up on. I'm going to go back 2 slides. You can see the histones here. The little green dots help wind up the DNA nice and tight. Now each half of a chromosome, part of this X, is called a chromatid. So we have a lot of words that sound similar. Chromatin is DNA when it's resting. We wind it up into a chromosome, and one of the pieces of a chromosome is called a chromatid. There are 2 chromatids that are stuck together and they're identical and they make up a chromosome. So when we go through cell division, the chromosome is going to divide in half. An identical piece of DNA is going to end up in each piece of the cell. A centromere is what is in the middle and it holds the chromatids together. Chromosomes. Now, good question here. What does chromosome number tell you about the complexity of an organism? The Adler's tongue fern is a plant, you can see it to the right there, has 1,262 chromosomes. A carrot only has 18. A cat has 32. Now, wouldn't you say that a cat is more complex than a fern? A chimpanzee has 48. A crayfish, much smaller than a chimp, 200 chromosomes. Earthworms have 36. Fruit flies have 8. Lettuce has 18. Sand dollars 52. Humans have 46 chromosomes. So what's the pattern, or is there a pattern? What does chromosome number seem to tell you about how complex an organism is? Hopefully when I asked that question you kind of went, well, it doesn't look like anything, and you would be correct. There is no connection, write this down, no connection between the number of chromosomes something has and how complex it is. Yes, a fruit fly is simple and only has 8 chromosomes, but isn't a sand dollar very simple and it has 42? Or, excuse me, it has 52? We only have 46. Why would a fern have 12,000 something chromosomes and we only have 46, and yet we think and feel and do things that ferns can't? So, chromosome number tells you nothing about how complex an organism is. How many chromosomes are in humans? Well, I just mentioned this. There are 23 pairs. You get a copy of each chromosome, one from mom, one from dad. 23 pairs for 46 total chromosomes. One from mom, one from dad. You can see in the picture here, we just number your chromosomes 1 to 22, and then we have an X and a Y. Chromosome number 1, you get 1 copy from mom, 1 from dad. Chromosome number 2, 1 from mom, 1 from dad, etc. A picture of the chromosomes, which you're seeing on the screen here, is called a karyotype. Scientists can actually take a look at chromosomes, take a picture of them, cut out the pictures, and sort them into which chromosome is which, and then lay them out in a picture. Very useful for finding diseases, because sometimes you'll see 3 chromosomes where 2 should be, or 1 where 2 should be, etc. Other important words that you should know. An autosome is a body chromosome. It has nothing to do with determining gender. So chromosomes 1 to 22 are autosomes. Chromosome 23, chromosomes I should say, are the X and the Y, and these are called sex chromosomes because they determine the gender of an organism. In humans, males are XY, females are XX, so this is a karyotype of a male because it's XY. Some other organisms work the other way. I believe in birds, females are XY and males are XX. But for now, just concentrate that in males for humans, XY, females, XX. Your next job is to complete the LO2 vocabulary sheet. So grab that from the crate, and if you're confused about anything in these notes, please come see me so I can clarify for you.